For today's episode, we will cover autism and Parkinson's. A major region of interest for both autism and Parkinson's is the basal ganglia. We've kind of covered the basal ganglia throughout a couple of episodes. Today, we will extend on the basal ganglia, especially the substantia nigra, which is heavily involved in Parkinson's and autism. We will also explore the direct pathway, the go area, and the indirect pathway, the no-go area of the basal ganglia. Now remember, the overarching identifier, or way you can think about the basal ganglia is, it's our go, no-go area. So the substantia nigra, it's deep inside the midbrain. It sits atop of the brainstem, essentially, sometimes called the tegmentum. Sometimes this is referred to as a region of the brainstem or near the brainstem. It's semantics. But the translation of substantia nigra is black substance or black matter. This is going to be huge here in the episode. It's black because of neuromelanin. Remember neuromelanin's roles, and it is black for a reason. I should say, Mother Nature uses neuromelanin here because it is black. It has special powers here. Because it is black, it receives all frequencies of light, the entire spectrum. So Mother Nature uses it here for this reason. Before I say the roles of neuromelanin, I also want to mention the locus coeruleus. Kind of the same scenario, same region as well, same little area in the tegmentum area of the midbrain. So the locus coeruleus is also black because of neuromelanin. And before I go into the details, remember the synthesis of tyrosine, because today we're going to talk a lot about dopamine and melanin, but also remember the locus coeruleus in the episode with the internal calculators and what makes autistics comfortable inside of ourselves. We covered an internal calculator in that episode, whereby the brain is calculating how much energy, effort is being used. Now remember, the locus coeruleus is the source of epinephrine and norepinephrine. You can think about it as brain adrenaline. So it's a very important role. So this calculator is monitoring how much effort we are using for the outcome. And this is what it's evaluating. Is the effort matching the outcome? And if not, a glia cell, an astrocyte. Glia cells are mostly known as support cells. And I think this, you can apply this here. Glia cells will come in and shut off the locus coeruleus. Hey, this outcome isn't getting you anything. It's coming in to save it. It's kind of like a friend. If you've ever had a friend or know of somebody, it's keep doing the same thing. It's just wrong. There's just a lot of effort being involved, but the outcome is not right. So you might have to come in and say, hey, what are you doing? I think that's what glia cells are doing here. So the synthesis of these comes from tyrosine, an aromatic amino acid. We've talked about that quite extensively. I think this is a hot topic. This is a fun topic. Aromatic amino acids are UV light detectors. So remember the 200 nanometer to 400 nanometer light. And tyrosine peaks at 280 nanometer light. This is absorption and emission we're talking about here. The strongest wavelength that arrives at the terrestrial land on Earth is 280 nanometer light. Pretty convenient there. This is the beginning of UVB, which is 280 nanometer light to 315 nanometer light. This is the strongest and the shortest wavelength of light. Now remember nanometers as well. There's 1,000 nanometers in 1 micron, sometimes called micrometer. And the thickness of a sheet of paper is roughly 80 to 90 microns. So this is how small we are talking here, and we need to go here. The reason I say that is this. Upstream, this is where we need to go. As small as we can, as upstream in the cell in our human biology as possible. Quantum biology and environmental signals. This is what drives biochemistry. How living organisms on Earth in the modern world are getting this light. But remember, light is how living organisms are on Earth. This is how we are created. This is the sources of energy. Cells receive energy, which provides energy to the living organism. Okay, so at the atomic level, how the powerhouses of the cells receive their powers. What powers the powerhouses? Cell responds to environmental signals. Remember the 2 episodes on mitochondria, the solo episode, and the episode with Dr. Richard Fry, where we understood the roles and sensitivity of mitochondria. And remember the episode on the cause of autism. We go into mitochondria there as well. So we're kind of talking about water being produced in the mitochondria, cytochrome C oxidase, and this being reduced, and this causes us a loss of energy. We need to consider that because this is huge for today's topic. If we want to think about what autism is, biological sense, a biological definition, autism is underdeveloped cells. It's a loss of energy. It's a lack of energy. Parkinson's is this process running in reverse. Now, dementias and these neurodegenerative conditions that are just sharp increase, quite frankly. This is taking a normal cell and unraveling it, losing energy. So the cell dies. The cell is losing energy. Now, what makes autism and Parkinson's so similar here? What makes autistics vulnerable to Parkinson's is we are already on pace of underdeveloped cells, cells burning out and losing energy. This must be understood. It needs to be considered. Cells die because they lose energy. Melanin drives transduction. This transfers energy from the environment into and through our biology. That's it. That's all you need to know about autism. That's all you need to know about the origins of Parkinson's. What's creating these conditions? If you can't go here, if you don't allow yourself to go here, you will not understand this. You won't understand anything with our modern health conditions. There is no power to these so-called powerhouses of the cell. And if you think about an effective fix, the treatment for Parkinson's is deep brain stimulation. This is it has a high validity. It has a high efficacy here. An electrode placed down into these midbrain regions, and it supplements the loss of energy. It's stimulating this, provides energy to the cell that we have lost because of our environment, the environment that we evolved under. And humans do this. Humans take ourselves out of the natural environment. We don't look at the environment as a whole from, let's say, 10,000 years, an epoch of 10,000 years, or 50,000 years, or even 200 years, or 150 years, or 130. As much as we want to narrow this down, the environment, our environment over the last 130 years has changed quite frequently and with rapid insults for our energy source. Okay, so the roles of neuromelanin. It's an antioxidant properties. Neuromelanin is likely one of our greatest defenses to environmental toxins, environmental insults. It binds metals, and this is a huge topic in current autism research. Environmental toxins. It binds to atoms and ions and makes it less reactive, less toxic, and less harmful, etc. So this is protecting the cell. Remember the sensitivity and the importance of the cell development. Other ways of describing this are sequestering. Neuromelanin acts as a sequesting. This also allows for detox of the cells, whereby it's a neuroprotection. Common and a region of interest for health is ROS, reactive oxygen species, redox. Okay, so a lot here with the redox. We cannot cover this, but ROS can be harmful to mitochondria, so the cells as well, and proteins and lipids and DNA. And also free radicals are huge here. Free radicals are an interest. We've covered some free radicals while discussing blue light and tech light, or any isolated wavelength of light, really. The primary region of interest here, the primary region of modern health is here. Next, neuromelanin is a neuronal regulator. Another easy connection, especially with dopamine here. Also, electrons with the energy sourced from earlier. What powers the powerhouses? Remember cytochrome C oxidase. Reverse engineer the ATP machine. This rotary engine that's pumping out ATP. Reverse engineer this. The step before is cytochrome C oxidase. Complex 4 that's producing water and free electrons. And then go down the line to the electron transport chain. And then to the TCA cycles and all these other cycles in the inner membrane that's pumping up enzymes and ions and so forth. Remember the chromophores in the mitochondria. The red light chromophores on cytochrome C oxidase and the VDR receptors. VDR are solar powered. Red light and the chromophores, of course, are solar powered. That's why they're chromophores. More and more on this light story. And humanity has seen rapid changes in light over the last 130 years. Match this with the rates of autism, which is challenging to do, I know, because we can't normalize. We can't compare rates from the 1950s or the 1980s even to today. There's just too much. You cannot normalize the data sets. But think about it. Are we using light the same way that we evolved under for millions of years, mammals? Or is light changed to be a convenience? So the question is, what do you think light is? Is it the same today versus evolution? This is so simple if you understand this. If you understand Jack Kruse's orange tree example, he provides, if you cover an orange tree up from the sun, if you block the sun, will it still produce the fruit? And even if the plant produces the fruit, will each orange be fully developed? Remember from the Dr. Richard Fry episode where I kind of use this analogy as well, and there's a short on it about causing autism to an infant tree. Trees have life stages as well. An infant tree, and it would be underdeveloped. We're taking a healthy tree and blocking it from the sun and accelerating its death. That's what we're doing with dementia. That's what dementia is. And if you understand that, it makes it a lot easier to understand this. So with neuromelanin and Parkinson's, there's a hypoxia of the pigmented neurons that break down melanin, the source of the power in electrons. Okay, so this creates a loss of dopamine, which is well researched in Parkinson's and likely the front runner of the cause. But let's go upstream in the biological path. The eyes, hair, and skin, these are the starting lines. Think about this in a linear path, a stack or a roll of dominoes. If the first domino misses the second, or if it just barely clips it, and everything gets off path, the process down the line is going to have dysfunction. There's going to be some implications here. Think about if it's not a source of breaking or falling and contacting, what if it's emission of power? Is the start signal that is not receiving power and it is responsible for taking power down the line? If the start signal isn't getting the adequate power, the process downstream is going to lose power. The farther downstream, the more implications. And that's what we're talking about. That's what we're always talking about. So the eyes here have melanin. And of course they do. You can think about the reason of the pupil being black for the same reason. And you can think about the RPE, retinal pigmental epithelium, and melanopsin. These are huge here. With this linear path, the goal is for this energy transduction. Okay, the source of that receives and absorbs the power. This transduction to remain efficient, powerful down the line, down this path. So to answer the question earlier, what is light? Melanin is chosen here because it is the dark pigment and it absorbs all frequencies of light. You understand that. We've covered it. In summer, go outside with the black clothes on versus white clothes on. That's not complicated to understand. We can make sense of that. Black is going to be much hotter, absorbing a lot more energy. This is why the pupil is black and this neuromelanin is black. Okay, so light is an electromagnetic field. This is the answer to the question, what is light? And it contains a wave and a particle, a photon. It's a wave-particle duality. Waves have the information. Photons carry the energy. Waves hit matter and transfers the energy. In the linear example of the photon, or photons, plural, hitting the first domino, the eye, skin, and hair, has to be. It must be powerful as possible to power downstream processes. We can think about some biophysics here as well, but I want to stay on topic here. Biologically, this is how we make dopamine. This is why dopamine is being synthesized through evolution. Serotonin as well, and proteins. Remember the episode on autism and gastrointestinal problems, where we talked about synthesizing these neurotransmitters and neuromodulators and so forth through the skin. The easy one that people can think about is vitamin D. So these things are photoreceptors, okay? This is happening. This is all across our biology. See the cause of autism episode as well for more detail. So artificial light versus sunlight is the cause. UV light wavelengths keeps the melanin and the dopamine optimized because it's receiving the proper amounts of energy. It keeps it optimized deep inside the midbrain too, where it is located. These little nuclei are not receiving the energy because they are downstream. The substantia nigra for the Parkinson's discussion is here. Tyrosine, an aromatic amino acid, remember, requires that UV light. Now, autism and Parkinson's, two things. There's two big causes here. One is a loss of or a lack of energy, and the other one is timing. Timing is huge because timing requires that UV light through the eyes, that monosynaptic connection to the hypothalamus. This is our master clock. This is what regulates all of our clocked timing. And later when we discuss the indirect and direct pathways, you might understand this. This might make more sense here. So also a form of synthesis here through the skin. And it's not complicated if you hear skin is the largest organ on the human body. That's not complicated. We're not going to dispute that. It's well established. But with monocytes, this is occurring on the skin as well with the monocytes or melanocytes. Melanocytes. Not white blood cells, melanocytes. Something we know in autism research is thyroid and the correlation with risk. Thyroid is tyrosine and iodine. This is well established. If you think about mitigating risk factors, if you know about this stuff, You might easily accept that a risk factor for autism is the prenatal or the mother having problems with hypothyroidism and also iodine. These two things are heavily supplemented in preparation for birth, in preparation for pregnancy. Iodine and the thyroid. So you might think about T3 and T4. This has strong data here, okay? Thyroid, it goes melanin to the SCN, to the hypothalamus. The SCN is in the hypothalamus, the master clock. The hypothalamus is also the body controller of hormones. So thyroid is going to be synthesized here. And the hypothalamus has direct connection to the pituitary. Pituitary just sits right below it. And this controls hormones as well. The pituitary is a source, a major region of interest for hormones. So we have T3 and T4. Remember, the 3 and the 4 represents the iodine. And T3, this is what I want to get into real fast. T has roles with neuronal functioning and motor functioning. This might not be well understood, or it might be easily glossed over, forgotten about, or not considered. But T3's role here with the brain development, fetal development. Okay, so T3 has roles with fetal development and through early childhood. And it's critical for brain growth. We know massive problems here in autism research about brain growth. Not complicated. It influences cell differentiation and then the migration. Remember, cells divide, they become specialized in the differentiation part. And then they migrate to similar friends, similar cells. In autism research, this is also well established. T3 also has roles with maturation of neurons. So contributing to the development of the neuron, which then implicates or satisfies, responsible for cognitive functions, neuronal activity for the T3, and it can regulate gene expression in neurons and affect neurotransmitter synthesis, receptor expression, and synaptic plasticity. All three of those are well established in autism research. Neuroprotector as well. T3 and neuromelanin have these neuroprotector responsibilities here. And T3's protection in the neuron has a lot to do with the ROS and aiding in repair processes and cell injury. And it's also responsible for metabolism and energy. Now, you might think about hypothyroidism. You might understand the downstream behaviors of this with low energy, mood swings, lack of cognitive function, difficulty concentrating, staying on task, so forth. But this is because if you go upstream, like we always should do, T3 regulates and affects the metabolic rate of the neurons, has massive influences on energy availability for the neuronal firing and maintaining neuronal health. This is huge. And synaptic transmission. And this also affects the gene expression. And the synaptic transmission and the fundamental for cell signaling. These pathways that we talk about quite extensively, such as the salience network, the default mode network, and the reward prediction layer, reward pathways. All of these connectivities with the basal ganglia is a good one. So we need to consider this. T3 has roles here, and T3 is shown to be implicated in both autism and Parkinson's. These are biomarkers for both. Now, you might think, you might ask, you ought to ask, T3, T4, this thyroid is a hormone. We're talking a lot about cells at the neuronal level. T3 acts to connect these. It is an interplay here with hormones and growth factors and neuronal activity. Dopamine has a close relationship with controlling hormones as well. This isn't that complicated. If you think about this stuff upstream and connect these pieces, all of these problems here that we're seeing with modern health conditions. Specifically here, though, we're talking about autism and Parkinson's. So remember melanin's role with water. The mitochondria is here, and the regions of the most neuromelanin are centered in the midbrain areas that we were talking about here, the substantia nigra and the locus coeruleus. There's a lot of neuromelanin here. Also, because there's a lot of water in the brain here. If you look at a picture of a brain, it kind of looks like a talon. Okay, there's a lot of water coming up through here in this area. And the melanin and the water are semiconductors. It's creating a lot of free electrons. The melanin and water interaction equals more electrons. And that's what we need. That's the source of our energy. Low electrons will cause low energy, a loss of energy. Okay, so earlier I said I would cover the direct pathway and indirect pathway of the basal ganglia. So we'll do that now. Remember the basal ganglia. It's our go, no-go area. Okay, so the direct pathway is going to be the go side. The indirect pathway is going to be the no-go side. Okay. So the direct pathway facilitates movements by promoting the execution of these motor actions. Remember the three kind of subtypes, the three considerations that we need to consider. The inputs of the basal ganglia, the relays, and then the outputs. Okay, so the inputs are the caudate nucleus and the putamen. This is receiving inputs from cortical areas, from the thalamus, temporal lobes, the midbrain even, with the substantia nigra for the dopamine. It's receiving a lot of inputs. That's not important. It's not exhaustive information that I shared, but it receives inputs and it's kind of facilitating based off of that. It's orchestrating the downstream behaviors needed. Receives inputs in the striatum. The dorsal striatum, remember, is the caudate and the putamen. And it receives excitatory information. So with the go side, we're going to use D1 like dopamine. And the caudate projects directly to the globus pallidus internal in this pathway. And the substantia nigra, the pars reticulata. Okay, remember the GPI and the SNR, globus pallidus internal, and the substantia nigra reticulata are essentially doing the same thing. They're pretty analogous with each other. Remember the globus pallidus as well while we're here. It has an external and an internal. And collectively, they form a triangle. There are two subtypes here. There are two subdivisions here. So the striatum speaks to the globus pallidus internal and the substantia nigra reticulata. Okay, it's doing this. It's quieting these two nuclei. If the striatum is quieting, shutting off the globus pallidus internal and the substantia nigra reticulata, that frees up information and activity in the thalamus. The thalamus is going to be the huge output receiver here. We're conducting these organizations, this downstream behaviors, orchestrating movements through the thalamus. It's leading up to communicate with the thalamus. So it's the striatum speaking down to the globus pallidus internal into the substantia nigra reticulata. It's inhibiting these two nuclei. And that frees up the thalamus to speak back up to the cortex and the motor neurons, sensory motor, etc. The goal here is to reduce the activity of the globus pallidus internal and the substantia nigra reticulata. Okay, and it's doing this with excitatory neurons. So this is the go side. D1 like dopamine. Remember the two dopamine receptor types. D1 like are excitation. DR1 and DR5. D2-like, which is the indirect pathway. These are inhibitories. So D2-like are DR2, DR3, DR4. So the indirect pathway, the striatum receives signals again, of course, and then the striatum is orchestrating. Okay, what downstream behaviors are needed for this environment? The indirect pathway acts more as a brake. Okay, the no-go side, of course. And the striatum inhibits the external globus pallidus. Okay, and the subthalamic nucleus, that other region of the basal ganglia. The subthalamic nucleus is essentially, in my opinion, the way I like to think about it is, it's the personal assistant to the thalamus. Okay, now, with this communication, it's activating. Okay, so the subthalamic nucleus excites then the globus pallidus internal and the substantia nigra reticulata, the things that the direct pathway shut off. Here they're being excited, and this inhibits the thalamus. So thereby it reduces cortical motor activation. This is important for fine motor skills and turning off movements. And in autism and Parkinson's, both have implications with the indirect pathway. The inability to shut off unwanted motor movements. If you think about autism, think about stereotypy and stimming, and even repetitive behaviors, even repetitive speech. That's another episode. But you have all of these repetitions and these sources of dyspraxia even, and the motor coordination and so forth. And Parkinson's is the same way. These are easy to observe in both conditions. So how to think about this is because dopamine's involved here. So we have a go side on a seesaw and a no-go side. And we're trying to conduct these appropriately based off of the goals of the living organism. So if we need to go, we can go. If we need to stop, we can stop. An implication here is the inability to stop. So that's why there's a strong risk for autism and Parkinson's. Essentially, autism is a biomarker or a precursor even to Parkinson's. This is going to be, in my opinion, what the data suggests, a region of interest. This is going to be unfortunate as more autistics are getting older. So with that, I think I covered everything that I wanted to cover here. The problem is connecting these things upstream to a common cause, which I don't think we're doing that great. And if you're listening to the podcast, listening to the episode, please feel free to leave a review or rating. In podcasting, reviews, ratings, and downloads are huge, and I very much appreciate your feedback. You can contact me on X at RPS47586, where we can discuss anything related to autism. You can check out the Hop link, so you can have links to all of the shows throughout different platforms of podcasting. You can check out the YouTube channel, check out the shorts and videos. They're starting to build up a bit. I guess you can check me out on TikTok. I decided to give TikTok a try. I don't love it, but we'll see how that goes. Thank you for listening to From the Spectrum podcast.